Have you ever looked at a plant and just been completely baffled by it? Well, today we're diving deep into one of botany's most beautiful puzzles, the 4 o'clock flower. It also goes by the name Marvel of Peru, and for good reason. A single plant can explode in this incredible riot of colors and patterns, which brings up a really fascinating question.
Right? How is this even possible? It seems to fly in the face of simple genetics. You've got one plant, one root system, and yet it's producing solid magenta flowers over here, solid yellow ones over there, and some flowers that are striped or speckled with both colors. It's a genuine mystery, and that's exactly what we're going to unpack today. And I mean, the level of variation is just staggering. We're not talking about subtle differences. You can see entire branches that decide to go with a totally different solid color. You'll find individual flowers that are literally split down the middle, half one color, half another. Then you get these delicate speckles on the petals, almost like an artist flicked a paintbrush at them. And if that wasn't enough, some flowers even change color as they get older. It's a living, breathing genetic mosaic. So, to get to the bottom of this, here's our game plan. We'll start with a little history of the plant itself. Then we'll look at the sneaky genetic tricks happening inside a single plant. After that, we'll see how pollinators get involved in the chaos. We'll do a really deep dive into the actual genetic code, and then wrap up with how you can cultivate this magic in your own garden. All right, first things first. To really appreciate this flower, you gotta know where it comes from. This isn't just some modern hybrid. It has a seriously rich history. That's right. Long before it ever showed up in a European garden back in the 1500s, it was being grown by the Aztecs. They valued it for both its beauty and its medicinal properties. And its scientific name? Mirabilis? Well, that's Latin for wonderful, a name it earned specifically because of this wild color-changing ability that has captivated people for centuries.
Okay, let's get into the mechanics of it. How in the world does one plant pull off this magic act? Well, the first part of the answer lies in things that are happening during the plant's own lifetime.
So the first mechanism is something called somatic mutation. Now, don't let the term scare you. The easiest way to think about it is like a tiny random typo in the plant's genetic cookbook. This typo doesn't happen in the seed, it happens in a single cell as the plant is growing a new stem or branch. And here's the result of that tiny typo. That one mutated cell divides and divides and grows into an entire branch where every single cell and every flower is following that new instruction. So if the plant's original recipe said make yellow flowers, this one branch with the mutation might suddenly have a recipe that says make red pigment, giving you this incredible splash of solid red on an otherwise yellow plant. But here's where it gets really wild. The second mechanism is something called jumping genes. And yes, they do exactly what it sounds like. These are little pieces of DNA that can literally cut themselves out of the genome and paste themselves somewhere else. When they land inside a gene that controls flower color, they break it, basically switching it off. But here's the kicker. They can also jump back out, fixing the gene and switching it back on.
And this explains exactly how we get those beautiful speckled patterns. On the left, a jumping gene has landed right in the middle of the recipe for red pigment. The recipe is broken, the result is a light-colored yellow flower. But on the right, as the petal grows, in some of those cells, that pesky gene jumps back out. The recipe is fixed, and those specific cells can now make red pigment. Since this happens randomly all over the petal, you get these gorgeous, unpredictable spots and streaks of red. So everything we've talked about so far explains what can happen within a single plant. But of course, plants don't exist in a bubble. So now let's zoom out and see how pollinators start stirring the pot, creating even more variety across a whole population. The 4 o'clock flower is actually the textbook example of a genetic principle called incomplete dominance. So let's say we cross a pure red flower, one that has 2 copies of the red gene, which we'll call big R, with a pure white flower, which has 2 copies of the white gene, little r. So what happens when they cross? Pretty simple. Their offspring get one gene from each parent, giving them a combination of big R, little r. But in this case, neither color is totally dominant. Instead, they blend. That one single dose of the red gene isn't strong enough to make a deep red color, so you end up with these beautiful pink flowers. But here is where the real magic of genetics kicks in. If you take 2 of those pink flowers and cross them, you don't just get more pink flowers. Oh no, the original parent colors come right back out. You get this perfect, predictable ratio. About a quarter of the offspring will be pure red,
half will be pink like their parents, and a quarter will be pure white. It's this constant genetic shuffling that keeps the whole population so vibrant.
All right, we've been using some simple analogies like cookbooks and jumping, but now it's time to get into the really deep science. Researchers have actually pinpointed the specific genes and molecular machinery that are making all of this happen. This quote right here is from a 2014 study that basically cracked the whole case. Scientists found the molecular smoking gun. They identified a specific jumping gene, a transposable element, sitting inside a piece of a color gene in the yellow flower tissue. But in the red spots on those very same flowers, they discovered that the jumping gene had been cut out or excised. This was the proof. And now we can actually put names to these things. The specific jumping gene is called DTMJ1, and it wedges itself into a gene called MJCP76 AD3. Now that gene is a critical piece of the biological assembly line that produces the red pigment, which is a type of chemical called a betalain. So when DTMJ1 is in there, the assembly line is broken. When it jumps out, the line works again. It's that simple and that elegant. So let's just quickly tie all of this together. What we first called a random color switch is, scientifically, a somatic mutation. Our jumping gene is a transposable element, specifically DTMJ1, and the act of it jumping out is called excision. And that beautiful mixing of colors we see across the garden bed, that's the work of incomplete dominance and allele segregation during pollination. See, it all connects. Now, the best part is that once we understand the science, we can actually use it. So this last section is for all the gardeners out there who want to become active participants in this amazing genetic story. You can literally play the role of the pollinator yourself. In the evening, right as the flowers are opening up, just take the stamen, that's the little stalk with the pollen on it, from one flower and gently dab that pollen onto the stigma, which is the receptive tip in the center of a different colored flower. By doing this, you get to decide which plants crossbreed, letting you experiment with creating new combinations for next year's seeds. And probably the biggest takeaway here is to embrace all that diversity. Don't just save seeds from your one favorite flower. You want to collect seeds from the solid-colored flowers, the striped ones, and the spotted ones to keep that gene pool as rich as possible. And hey, if you find one pattern you just absolutely have to have again, you can actually clone the plant by dividing its tuberous root in the fall. That preserves its exact genetics for the next season. You know, the story of the Marvel of Peru is just this perfect, beautiful reminder that genetics isn't always neat and tidy. It's dynamic, it's a little chaotic, and it's absolutely full of hidden potential. And that leaves us with one final thought to ponder. What other incredible genetic dramas are quietly playing out, just waiting for you to notice, right there in your own backyard?